Howdy everyone, you're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a PhD program while trying to stay sane. We also invite faculty from our interdisciplinary program here at Texas A&M onto the show to pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M. In this episode, we'll be talking to the speakers of this year's Genome Editing Symposium here at Texas A&M. They work in various sectors from academia to industry to government, yet they all use their knowledge of genetics on a day-to-day basis. Today, we hope to get more insight on how our listeners can apply their degrees post-graduation. My name is Anthony, and I'll be one of your hosts today.
And my name is Joe. I'll be your other host today. And we'd like to start off by thanking you both, Dr. Barrick and Dr. Saunders, for coming to the Texas A&M Genome Editing Symposium and agreeing to be on the podcast. It means a lot to us. But we'd like to get started maybe with just a brief introduction. If you guys could tell us a little bit about yourselves and maybe your research that you do.
Yeah, my name is Tom Saunders, and I classify myself as a genetic engineer. I work in the Transgenic core at the University of Michigan. And my job, my research, is to make genetically engineered mouse and rat models of human disease for investigators to use in their research programs so they can better understand human health and disease by testing out ideas in animal models.
Great, and I'm Jeff Barrick from the University of Texas at Austin, right down the road from you. I work in the Department of Molecular Biosciences there, and my research really has its basis in experimental evolution, understanding genetics and systems where we can watch evolution play out. But recently, I've started working on genetic engineering and synthetic biology of microbes, especially ones associated with insects like the honeybee, and thinking about some of the applications and also interesting evolution and ecology in those kind of communities of microbes.
Cool. So we got two pretty different research areas, but united by genetics and gene editing and genome engineering. So,
can you guys give us some background on where you got your PhDs from and in what field?
I earned my PhD at the University of Texas Health Science Center in Houston, Texas. My degree is in biomedical sciences with a concentration in immunology. And just as I was wrapping up my PhD, the field of DNA cloning was being born. So my postdoc, that's what I went for, was to learn more about molecular biology and how to clone genes. And then just as I was wrapping that postdoc up, what came along was polymerase chain reaction. Polymerase chain reaction was a game changer. It changed science. And if you look at the number of papers published that included polymerase chain reaction technology, it skyrocketed back then. Just in a few years, it took over. And so that's where I got my start in molecular biology. And then I went from there into working in a transgenic core laboratory where our goal was to make transgenic animal models by inserting foreign transgenes into chromosomes of mice, typically by random integration, and then you have a genetically modified mouse. So I can pick up with some interesting mouse models in a bit, but I'm going to let Jeff talk about his start.
Great. So I kind of found my calling in science, or the field I was interested in early on when I was a freshman at Caltech, and I heard somebody talk about doing evolution of molecules in the test tube, where you could kind of turn cycles of selection and replication. And so I've really been interested in applications of evolution since then. And so I've moved a little bit to more and more complex systems, from test tubes full of molecules, RNAs and proteins, to working on bacteria and their gene regulation, to working on model systems where you have defined communities or single microbes in the lab and you can watch them evolve for a long time, up to now communities of microbes living in natural environments and microbiomes. And so I've enjoyed interacting with all kinds of people along the way there. My PhD was at Yale University, and then I worked at Michigan State for my postdoc, and just to really follow the science along the way.
Great. So there's there's a number of fields you can go into with your PhD, especially a PhD relating to molecular biology, genetics, things of that nature. What made you both choose academia, and what was that route like? You explained a little bit about your PhD, but were there any defining moments or experiences that y'all had that really pushed you into that career path?
Well, for me, it was gaining experience in mouse models. So in my PhD, I worked on a mouse model of cancer. Then in my postdoc, I worked on a mouse model of human osteogenesis imperfecta type 1. And I wanted to keep going, so I had experience in mouse models. And then I had the opportunity to work with Sally Camper to set up a transgenic core facility at Michigan. So I seized that opportunity, and then I realized that in my boss, Sally Camper, I had an outstanding human being to mentor me and to coach me and to let me develop. So I ran with it, and now I'm an expert in the field and recognized by my research community as an expert in the field. So I'm not, world famous or anything, but my colleagues appreciate that I can have conversations with them, give them input into ideas on how to approach genetic questions. So today I had the opportunity, thanks to the organizing students of the symposium, to meet with Dr. David Threadgill. And we sat down and had a nice talk about his ideas for a new mouse model for his lab to pursue research in with his graduate students. And so I threw some ideas around with him. So maybe he'll incorporate some of those into his studies, and his students will carry that forward and study them.
Yeah, so I think, we're of a much older generation than you, where we didn't think so much about careers maybe going in and those possibilities, and there weren't so many symposia and different kinds of speakers coming and talking to you. So I'm quite happy with being in academia, because it's kind of like running a small business, running a lab in some ways. It's really intense. You're working really hard. You really get to know the people around you, mentor them in your lab. And there's a lot of variety in what you can do. So we teach, we get to interact with students in all kinds of different environments. We get to do outreach. And I really like that variety, and I kind of value that, in addition to being able to explore new topics and kind of shift over time what I've done after I talk to people and get excited about what they're doing. So I feel pretty lucky and privileged to be in that kind of environment. And a lot of my students are going off into very different environments and having a lot of fun in industry, and synthetic biology is kind of having a heyday right now, where there's a lot of, almost Google-like companies where you get to do some exploration and work with all kinds of great machines, automation, all these kinds of things I can only imagine out there.
So I guess going off that, how do you both find mentoring students that want to pursue something outside of academia? How does that give you any obstacles when you're trying to help them find that next step, since you don't really have as much experience in that field?
Well, actually, since I'm primarily a technologist directing a core facility, I do not train up graduate students, so I don't technically mentor students in the same way that Jeff does. So for me, I bring in the students and we give them a short experience in the lab working with transgenic technology or CRISPR technology or embryonic stem cell technology, and it's kind of like a boot camp, a short, defined, intense experience where they get exposed to a lot of ideas, a lot of scientific principles that they can then take back to their own lab and apply. And we're there to help them out. When they have questions, they'll come back, bounce ideas off of us. But I don't have the same level of formal direction as Jeff does in mentoring. So I think he's better qualified to answer that question.
Yeah, so I've seen a complete reversal when I go and give a seminar places and people talk about like where they're thinking about going from being like 90% academia to 90% industry over the past, decade or so. And I'm actually kind of comfortable with that, and I tried to educate myself about those opportunities. In fact, one of the last times I was here on this campus was for an NSF I-Core program where professors and one of their graduate students or postdocs go out and kind of explore a possible idea related to your research that could be, found a small startup company and things like that. And so I got to go and do that and then interview a ton of people at companies, including all kinds of alumni from my program. And so some of my students, one of them does business development at one of these companies. And so he's always willing to talk to these people about ideas and paths. And so I don't necessarily mentor them personally myself at that level, but I have all kinds of connections I can give them. And I'm super excited to hear about what they're doing and learn about what's going on.
I think those connections are really what's important in industry or in academia, is making those connections with people in your field, and just networking essentially is one of the biggest things that you can do as a grad student to help you prepare for that next step. I think I've seen that across the board here.
Yeah, and even if not for, an opportunity for a job in the future, a collaboration, if there's some research overlap, or somebody that, you trust their critical thinking skills and, their eye to look over papers and maybe review some of your manuscripts and stuff like that. So absolutely. Okay, so we talked a little bit about some of the research that y'all both do. I'm sure that you have a lot of projects going on in your labs and at your core facility. Is there any one particular project that you're especially excited about to talk about, whether it be at the symposium or just something that you have been thinking a lot about, a project that's been exciting you?
Take it away, Jim.
Sure. So tomorrow I'm mostly going to talk about work doing genetic engineering of bacteria that live in the honeybee gut. And that works at a fairly advanced stage. So I'm going to talk about something at a less advanced stage that I'm kind of excited about, because that's the way we are, right? Always looking to the next thing. And as I've come in from an outsider and learned more about entomology and also microbiology and much more complicated environments that I'm used to thinking about in simple microbiology in the lab, I've become really interested in how microbes become symbionts or endosymbionts of insects. And we have kind of a system where we can culture some bacteria, and they're related to some bacteria that live inside of aphid bodies and cells and are passed along through generations. And I'm really excited to think about the combination of either using some directed evolution plus some engineering to try to get a microbe to take that step where it transitions probably from something along the lines of an opportunistic pathogen, something that's in the wrong place and exploits the environment it's in, being the insect body and eventually killing the insect, to something that can be passed along in, if not mutualism, at least symbiosis stably over time, a process where you can look back and you can see this has happened by seeing what's out there in the diversity of life. But I really like to see if that process happened and see if we can push that process along a little bit. So I'm excited about some of that. For one of my newest students is kind of taking that project on.
So in my lab, what we're pushing is more efficient ways to make transgenic mice and rats. So when CRISPR happened, that gave us the ability to do precision gene targeting. And that was a real disruptive breakthrough in the field. It was game-changing, again, just like PCR had been. And so now the question is, how can we fine-tune that? And so currently what we're chasing is a genomic landing pad in a specific chromosomal location with a new technology. Well, I don't know if it's new, but a different approach. So we use CRISPR to engineer a landing pad in a defined location, and within it we place DNA sequences recognized by BXB1 integrase, with the idea that the BXB1 integrase then will simply exchange whatever is in the original landing pad with new genetic information that lets the CRISPR reagent, which we have embedded in that same landing pad, target a different gene. So it's like you just switch it out, and that's more efficient than creating a new animal model every time. You just switch out one piece for another small piece, and you've got a new target, a new animal model. So that's what we're chasing. We're going to see if we can set that up and apply it to cancer-causing mutations in the TP53 gene, a known oncogene involved in human cancers. So that's the one that we're chasing down now that I'm excited about. And, I could keep going. Yeah, so I'm going to stop there.
We actually do a decent amount of, we call it having like a transgenic docking line, but we do similar experiments with mosquitoes when we want to insert different transgenes, but we want to maybe just change small things. So we might have a strain of mosquito that expresses CRISPR, Cas9 enzyme in the germline, but we want to insert a different guide RNA to disrupt a different gene. So we have an integrase or a recombinase sequence that's inserted within that transgene, and we can kind of swap out and insert guide RNAs. I guess I'm curious, whenever you do that, if you swap out that cargo for that transgene, every time you're going to change the sequence, like you said, you don't have to make a totally new transgenic animal. You can just kind of go back into that one and treat it with the recombinase and whatever sequence you want to insert. Do you have a new selectable marker every time so that you know that the new sequence has been inserted?
Well, that's the beauty of using CRISPR. We can directly modify the genome in fertilized mouse eggs without using a selectable marker. Before CRISPR, we would modify genes in the genome in mouse embryonic stem cells, and that required the use of a selectable marker. However, early on in the field, it was recognized that the presence of the selectable marker and its promoter on the chromosome was enough to express gene expression, not only of the gene you were targeting, but upstream of downstream genes. And these could produce a phenotype that you weren't expecting by the presence of the selectable marker. So today with CRISPR, we can avoid the use of a selectable marker, and we can avoid the use of a technology, for example, FLIPO recombination, and the requirement to remove the selectable marker to have a clean model. So that's one of the nice things about CRISPR. And so I'm waiting, So with Cre recombinase, the most interesting thing we've done lately is applied co-inversion with heterotypic LOX-P sites. So like LOX61 and LOX71. And as I was discussing with Anthony last night, we can place a coding sequence in a gene intron backwards, so the protein is not made. But if we flank it with these heterotypic LOX sites, and then we express Cre recombinase in a cell-specific fashion, then a subset of cells, we can turn that sequence over and now express the protein in a controlled fashion in a specific cell type to simulate a human disease condition or two.
It's almost like
make the model
switch.
Anthony wants to study so he can earn his PhD,
Right.
publish that big impact paper, and get his first grant.
No, that discussion was really helpful last night, 100%. And going off that, I guess you both are very good at communicating your research to the layperson, someone who doesn't understand it. And you have to do that at these symposiums, right? When you're giving these talks. So what kind of advice would you give to our listeners who are preparing that first poster presentation or that first 3-minute thesis that they have to go out there and try to disseminate their science to people who might not 100% get it?
I think that's challenging, and I see it in my own students that I'm mentoring. It's very challenging to take yourself outside of all the details after you've been in them so long. But I think just remember that it's a story that you're trying to tell, and that people are interested a little bit in how you got there, but you have to simplify things so much, right? You need to simplify. I'm actually mentoring an iGEM synthetic biology team, and they're making a website and trying to tell their story right now. And you just have to remind people to simplify things down. I think the practice you get in academia teaching classes actually is really helpful with this. So maybe take advantage of that when you're TAing classes. Maybe give a few, side lectures, or your recitation section, practice some of those skills too.
That's a good point.
Yeah, for the genetics program, we actually have a 2-semester TA requirement for the genetics lab for undergraduates. So I've noticed even from the 2 semesters of TAing, but also we have some programs where we can mentor teams of undergraduates. That's helped out a lot. And we have committee meetings every year, and that's one thing my committee, I think, says, or has been saying, is mentoring undergraduates, explaining the research, kind of at different levels to your committee members, and trying to go to the highest level of expertise, and and say everything accurately, and include all of the necessary information, but also being able to kind of make it a little more concise, summarize it up for an undergraduate team as well.
Yeah, so I agree with Jeff. You have to be able to tell a story. If you're just going to give a recitation of facts, the audience is going to fall asleep. So if you can tell a story and get people to follow along by keeping it simple and not going into a ridiculous amount of detail, like reciting the DNA sequence of a LOX P site.
Right. Right.
Then the audience will be able to follow along. If you can give them the big picture, let them know where you want to go early in the talk, and then take them along the way, step by step, until you get to the answer and tell them the story. And then, don't give them the conclusion at the beginning, give them the conclusion in the middle, give them the conclusion at the end, and by the time they hear it the third time, they may cotton on to it.
Yeah, and we're certainly interested in the research stories that you guys have to tell tomorrow. But I guess we're also curious about, we talked a little bit about your career path kind of story and each step that got you to your current position where you both are at. I'm curious if there are any obstacles that y'all have faced during that time period of getting to the director of a transgenic core or a professor at the University of Texas at Austin, and how did you overcome that?
I can take that in a few different contexts. I think one thing that I did during graduate school was I came in and I was like, I know exactly what project I want to do in exactly which lab. And I rotated in some other labs, and it was fun to learn about the stuff, but I enjoyed that lab in the end. And then I worked on that experimental project through the entire course of my PhD, and it never worked. Yeah. But I was paying attention to what was going on alongside me and helping a lot of my colleagues and learning other skills. And so I think the obstacle here was myself a little bit. And I really wish that my PhD advisor had taken me aside and said, are you sure you want to keep concentrating on that thing? Or maybe, rotate fully. And I wrote my thesis on this other topic, right? Not my experiments that were not working. So I think you have to be a little bit flexible. Technologies will change. The expertise around you is really important for getting your own expertise. You need to ride the wave of what's working. You can return to your kind of true north. And, when I switched to my postdoc, I switched back to working more on these evolutionary topics related to what I had been deflected to, but really enjoyed working on during my PhD. So I don't know if that's an obstacle, or maybe that's a way to think about the process is you have to be a little bit flexible sometimes, not too flexible where you're changing what you're doing every single time you encounter some difficulty, right? But working with the community around you to be successful.
Troubleshooting is the story of a PhD. Troubleshooting and pivoting different projects.
Always talk to someone who has done the technique with their own hands. Never try to learn something by reading papers, right?
That's great advice.
Yeah, I would have to agree with that. So you're starting out in your PhD, you've qualified, you feel confident that you know what you're doing, and you may be tempted to not ask for help. So that means you should be asking for a lot of help from a lot of places, because people actually like to help you. They want to give you advice. And if you can pick that up on that, take that advice and use it to your advantage, you'll find that you'll make a lot more progress than if you're stewing in a lab trying to figure out how to run a gel for the first time or set up a complicated piece of equipment for the first time. Asking for help, getting help, and then using that information is a key to success.
So now that we've talked about some of you guys' obstacles, any highlights over the past career?
Yeah, so I got a couple I can throw out there. So one highlight was working on a deaf mouse model.
Okay.
So this is a spontaneous mutation in mice. So back, in the hundreds of years ago, the Japanese emperors, they would keep pet mice that would twirl around in the cage because they had an inner ear defect. They didn't know which way was up. So they would just always be spinning around, and they're very entertaining to the Japanese emperors. So they're called twirling mice. It was a spontaneous mutation. Nobody knew what a gene was, or genetics, or inheritance. Oh, look at this squeak. And so, so they archived all these mice live at the Jackson Laboratory for years. And then they discovered DNA and chromosomes and inheritance and all that jazz. And then they discovered they could map genes. So I was working in the lab with Sally Camper, and she was looking at this mouse. Why don't we map that gene? Oh, okay. And so with a couple of students in the lab, they did the back crossing, the breeding of the mice to try and isolate out which part of which chromosome mapped to that behavior, to that twirling mouse. And they mapped it down to a chunk of the chromosome. It's like, okay, what do we do now? And so about that time, right, this is just before the human genome and the mouse genome was sequenced, right? That was like, 2000? A huge landmark for science. There were these genetic chromosome libraries. So we could just order up a chromosome library and pluck out, we want this particular book in that library that matches the place we mapped in the mouse genome. And so we did that. Now we've got these two. They map really close. But which one is it? Is it A or is it B? Who knows? And so the student in the lab, he prepared DNA from chromosome fragment A and chromosome fragment B. And he said, Tom, here's the DNA. Oh, okay. And Tom, look, here's a mouse colony, and we're going to make fertilized mouse eggs from these crazy, deaf, twirling mice. Oh, okay. That's just the hands. Come on. And so I collected the mouse eggs, I put them on the scope, and but we didn't know which DNA to inject, right? So I started injecting prep A. And DNA prep A, oh man, this is crap. It's blocking my injection needles. I can't get it out of the needle. Screw it. And, so I go to prep B. So I inject, it works pretty good. So I inject it, right? So completely random, A or B. And the other thing is, to make fertilized mouse eggs, you need mice. We didn't have a lot of these crazy twirling mice. So I had a very limited number of eggs that I could micro-inject. And then we only had 8 pups that were born, right? So I'm sitting there, okay, what's going on? And so then the graduate student crashes into the lab. Tom, you gotta look at this. We go and we look at the mice. And so there's a litter of mice, right? So now they're about 2 weeks old. So they're old enough to stand up and move around. And so 7 of these pups are swinging around, doing that crazy, no inner ear function twirling dance. And there's one little pup sitting there in the corner just looking around at all these crazy mice. And he says, that's the mouse. And he takes a little snip of the tail tip, extracts the DNA, does the analysis. Yes, it's in there. And I say, great, what's on that piece of DNA? And, I don't know. How big is it? It's only 150,000 base pairs. And it's like, he looks at me and I look at it, 150,000. Yeah. So at the time, if we did a gel and we could read off 1,000 base pairs, that was a big deal. And now we have to do this 150,000 times. So I look at him and he looks at me and says, you got to. So I tell him, call a company up and see if this company will do it for us. So he calls the company and oh yeah, we can do that. It'll just take 6 months and $250,000. It's like, so if you're an academic research lab, do you have that kind of money? No. So our boss, Sally Camper, what does she do? She calls the NIH. What does the NIH have? They have a human population of patients from Pakistan with, guess what? Hearing impairment. And it maps to this spot in a human chromosome, which is equivalent to the same spot we're studying on this mouse chromosome. So she says, I bet if we find that's the same gene in mouse and man causing this mutation, and we found it, it's on this DNA fragment, you're at the NIH, you've got government dollars, why don't you sequence this for us and find out what it is? And so the guy, Tom Friedman, said, that's a great idea, I'll do that. So he did it. And sure enough, there's this, it's myosin 17, I believe, was on that mouse fragment. And they looked in their human patients, and that same gene was disrupted in the human deaf patients. And so we published the 2 papers back to back in Science magazine, one for describing the mouse discovery and one discovering the human discovery. So that was an exciting piece of research that I contributed to, right? But it was a whole team of people. It wasn't just me. I mean, So that's the other thing, if you're going to be successful, you're not going to do it sitting in the corner of your lab with a couple of technicians or a couple of students. You need a team bringing together different expertise to achieve that project goal. You can't just sit there in a corner.
Yeah, that goes back to the collaboration that we were talking about earlier, about how I think especially here at A&M and in our interdisciplinary genetics program, collaboration is a big part of the research that gets done here. So I think that is a great point to say that your successes aren't only yours.
Yeah, that's great. I mean, to pick up on that theme a little bit with my story. So I think maybe my most surprising or, interesting success was maybe in graduate school. And so the thing that I did end up working on was that when I had arrived at the lab, they've been studying little DNA enzymes that cleave themselves and gave a signal when they bound to a small molecule. And I was like, oh, that's cool, piece of biotechnology, maybe I'll work on that. But what they eventually realized was that this was going on inside of cells where pieces of RNA in bacterial cells were folding up, recognizing molecules like amino acids, vitamins, and they were turning on or off the gene that they were next to. They were doing autoregulation, but it was the RNA with no protein involved, which at that point it was not really known much that this could happen in bacteria. And so I was super lucky to be the kid who had been coding since junior high and who could do the bioinformatics and look for these conserved RNA structures next to genes of similar function, put the pieces together and say, oh my gosh, that must be binding to, adenosylcobalamin, which is vitamin B12, or binding to guanine because it's involved in these genes. And there were just so many of these stories going through one and another, and I got to collaborate with everyone in the lab. I got to do really exciting things like go and work at the Sanger Center in England with their database, the RFAM database, where they have these RNA families. They use them to annotate genomes, work with those people for a month based on this, because I learned that I was the person submitting the most RNA families ever to them at the time, right? So they're like, well, why don't you just come and we'll watch what you do and we can interact with you. So that was a great opportunity. And then maybe the highlight was that we wrote a Scientific American article about this. And, I had grown up reading that magazine and I thought that this was just really cool and something I had never imagined I would have the opportunity to do. But I literally was out to dinner with my we were recruiting some postdoc and my PI was there and I was joking. I'm like, when are you going to write your Scientific American article, Ron? Ron Breaker was my advisor there. And he's like, he got very serious. And he's like, I should get back to them on that. And I'm like, oh, I'll do it. I volunteer for this. So that was just super exciting. I mean, this process has played over and over again a few times throughout my career in different ways, and there's other exciting things too. But I think that's really special to me, and
it's a very cool full circle moment.
Yeah,
you've been growing up.
And again, it was working with the right people and being able to help them out and use my skill or background in a way that added to what everyone else was doing.
Okay, so we have some speaker-specific questions for y'all. So I have a question for Dr. Barrick. So engineering microbes has been a pretty hot topic, as of lately in research. And I guess more recently, I heard a TED Talk of Jennifer Doudna, and she was talking about engineering the microbes within the young livestock gut to try to inhibit methane production and emission. I'm curious, I know that you do some microbe engineering and endosymbiont for honeybees. Can you think of any other applications where engineering microbes could be really beneficial, whether it be for society or for pushing the field of science and just basic research forward, or any way that it could be beneficial?
Oh, wow, that's a huge question. I think that I want to say one thing, which was it's a related question, is will we ever be able to use engineered microbes in applications like that? And how is it regulated?
That was actually my next question.
Yeah, and so one thing I've seen in visiting some of these companies that are working with engineered microbes is this parallel paradigm where people will go and they will search all of biodiversity, get a bunch of isolates and test them all for when we paint them on seeds and plant the seeds, do the crops develop better, or are they protected against pathogens, which is mining natural biodiversity. Yet the alternative of doing the genetic engineering on the microbe to accomplish it is completely disallowed. You're not allowed to put that microbe out there under most circumstances. And there have been very few studies looking at these things in complicated systems and how the genes get disseminated or the microbes get disseminated. So I just see that as a really interesting contrast where you can put gobs of some microbe in some new environment, but you can't put a new gene in a new environment, right? And I don't know what the right answer is for some of this stuff. Obviously I want us to be safe and keep containment of things and understand what's going on. And there's going to be different answers in all kinds of different systems. In terms of what I'm really excited about, I obviously would like to keep biodiversity out there, but maybe block some of the pathogens that some of the biodiversity is spreading or prevent those transmissions. So, aphids are one of these things where aphids are not so bad for a plant, but they spread plant viruses, which are really bad for a plant, right? So if you could block that in some way without disrupting other things, I think that would be really interesting. And then there's the whole realm of human living therapeutics, right? Engineering microbes that you would take as a pill and they would maybe reside in your gut. And medically and kind of how people make money in medicine is completely at odds with how some of these things might work, right? One paradigm, the safe paradigm, is to make cells that cannot replicate in the gut, right? So you would take them all the time and they would just be living chemical factories. And maybe that's the way to do it. But the alternative, that doesn't solve the problem for someone's lifetime, right? If you could implant that microbe, if you could make sure that it's genetically stable over time, the genes, and make sure that there aren't going to be any bad consequences, a lot to ask, a lot of experimentation would be necessary for that, then you could potentially, give them support for a missing enzyme in their metabolism throughout their life, right? So I think it'll be a really interesting time to compare these things. And I'm not sure that there won't be some other solution that some new technology comes out with that, maybe makes some of these worries not even relevant. But it's interesting to think about.
Absolutely.
And to ask a similarly technical question to Tom, when you are creating these transgenic mice, how do you screen for things like off-target activity of CRISPR? I know that's a very big issue that people that use CRISPR are looking at. Is it affecting where we want it to affect?
Yeah, so off-targets are an important consideration. So we use something called Hi-Fi Cas9 enzyme from a company, and it's an engineered protein that reduces the number of off-target hits. And the other thing is there are bioinformatic tools, algorithms, that will predict where off-target hits will occur in the genome based on the DNA sequence. So we can easily PCR amplify those sequences and take a look at them at the DNA level to see if there are any unusual mutations. The other thing, when we design our protocol for designing these genetically engineered mice, is we're going to select reagents that are unlikely to target a gene. So if we screen a guide RNA that will direct Cas9 protein to make a chromosome break, we ask, are there any possibility for an off-target hit inside a protein coding gene? And if there are, then it goes in, we don't want to use it. And then if we find another one and all of its possible off-target hits are in enterogenic regions, like 300,000 base pairs between gene A and gene B, the probability that's going to disrupt protein function is minimal, so then that's less of a concern, and we'll go ahead and use it. And so it's called genetic screening.
Right.
Today's episode was hosted by myself, Joe Romanowski, and Anthony Matarazzo, and produced by Brittany Ming Whitfield and Emery Alexander. This podcast is brought to you by the Texas A&M University Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, Twitter, and TikTok at Genetics Podcast. Thank you for listening. We hope you'll tune in next time.